Welcome. We are in the middle of a pandemic. It has been affecting the lives of everyone around us. There is a race to find a vaccine for COVID-19. Maybe this is the time to look at the story of vaccines. This topic is relevant from the point of view of BS Paper 3 under Science and Technology. In this video, we will discuss what is a vaccine, what are different types of vaccines, future development in the field of vaccines, how do the vaccines work, vaccine development process, and we'll see the present status of COVID-19 vaccine development race. A vaccine is a biological preparation that provides active acquired immunity to a particular disease. Vaccines are like training courses for the immune system. They prepare the body to fight diseases. A vaccine stimulates the immune system to produce antibodies. Vaccines harnesses the natural activity of immune system. Vaccines contains the same germs or part thereof that causes diseases, but they have been either killed or weakened to the point that they do not make one sick. Vaccines are designed to prevent disease. The concept of vaccine has been around for centuries, but a British physician, Edward Jenner, is generally credited with ushering the modern concept of vaccination. Jenner, in 1796, used metaform cowpox postulate to inoculate patients successfully against smallpox, and by 1798, first vaccine against the smallpox was made. After Jenner, Pasteur's rabies vaccine 1885, live attenuated cholera vaccine 1897, and inactivated anthrax vaccines in human 1904 were significant achievements. Now let's have a look at the types of vaccines. By the antigen used in their preparation, vaccines may be divided into live attenuated vaccines, inactivated vaccines, toxoid vaccines, and conjugate vaccines. Live attenuated vaccines. It contains the whole bacteria or virus which have been weakened. Live vaccines tend to create a strong and lasting immune response and are some of the best vaccines. For example, chickenpox, rotavirus, yellow virus, etc. Inactivated vaccines. They contain whole bacteria or virus which have been killed or part thereof, like protein or sugar. It do not create as strong and lasting immune response as live vaccines. They usually require repeated doses and or booster doses. For example, inactivated polio vaccines, hepatitis A, toxoid vaccines. It uses toxins made by the germs that causes the disease. Here, the immune response is targeted to the toxin. Tetanus toxoid and diphtheria are some of these examples. Conjugate vaccines. Conjugate means connected or joined. In the early days of polysaccharide vaccines, it was found that they were not working well among young children. Researchers discovered that if polysaccharide was attached with something that they knew created a strong immune response, they worked much better. In most conjugate vaccines, the polysaccharide is attached to diphtheria or tetanus toxoid protein. For example, hepatitis B. Remarkable research and development is happening in the field of vaccines. The focus is to make it more safe, cost effective, efficient, and personalized. Let's see some of these remarkable developments. Nucleic acid based vaccine. Here, cells are injected with a genetically engineered plasmid which contains an antigen to cause an immune response. It has the potential to induce wider range of immune response. But currently, no DNA vaccines have been approved for human use. mRNA vaccine. These are very significant, especially from COVID-19 point of view. An mRNA sequence is coded for a disease-specific antigen. Once produced within the body, the antigen is recognized by the immune system, preparing the body to fight for the real thing. RNA vaccines are faster and cheaper to produce than the traditional vaccines. There are two types of mRNA vaccines: conventional mRNA and self-amplifying mRNA. Self-amplifying mRNA vaccines generate more antigens, resulting in a robust immune response. The mRNA 1 to 700 vaccines of Moderna, which is currently under phase 2 trials, is a promising candidate for COVID-19. Recombinant DNA platforms. It produces an exact genetic match to proteins of a virus. The protein's DNA combined with an DNA from a virus harmless to human, and once injected, it will produce antigens which will stimulate the immune system to protect against the virus. Virus-like particles based vaccines. VLPs are molecules that closely resembles virus but are non-infectious because they contain no viral genetic material. Now, VLP contains high-density display of viral surface proteins that present an antigens, which results robust immune response. Non-replicating viral vectors. Viral vectors are tools used to deliver genetic material into cells. A viral vector vaccines induces expression of pathogen proteins within the host cells, similarly to attenuated vaccines. As viral vector vaccines contains only a small fraction of pathogen genes, they are much safer. Recombinant nanoparticles. Recombinant DNA is artificially created DNA. This type of vaccines uses genetically engineered three-dimensional nanoparticles that incorporates recombinant proteins critical to disease. In short, they support production of antigens that causes an immune response. Peptide vaccines. It uses synthetic proteins that mimics naturally occurring proteins from a virus, causing an immune response. Now, let's see how do the vaccines work. Vaccines mainly adopt four different methods. All the methods rely on antigen presenting cell APC to initiate the immune response. The APC identifies peptides or snippets of virus proteins, and immune system will then be enabled to neutralize the virus. In order to understand how do the vaccines work, it helps first to understand how our immune system works. There are 100 trillions of bacteria and viruses in our body and all around us. But not all of them causes diseases, but some can. There are natural barriers to stop them, but sometimes some of these disease-causing bacteria do get through all the barriers. The immune system recognizes them as foreign invaders. But how the immune system recognizes them? The immune system recognizes them because the proteins or sugar on the bacteria surface have different shapes to any of the ones in human body. For example, here you can see four different viruses. The immune system recognizes these characteristic structures. This triggers a complex chain of events involving many different types of white blood cells working together. One type of white blood cells is able to make antibodies to fight the invaders. The antibodies can stick to the proteins or sugar on the bacteria surface, and this kills the bacteria or disables them. However, there is a catch. Not all antibodies will work against these bacteria. They have to be exactly the right shape, a bit like a key fitting a lock. Our bodies have a library of billions of white blood cells, each of which can make just one set of antibody. Only a few of these antibodies will match the invading bacteria. But producing antibodies of the right shape can take several days. By this time, the invading bacteria would multiply to billions of disease-causing bacteria. Once the right cells are activated, they quickly divide and turn into production line, making masses of antibody that stick to the bacteria. Eventually, our body gets rid of all the bacteria. Antibody remains in the blood. Some white blood cells may also become memory cells. And if that specific bacteria invade the body again, the immune system will respond so quickly that one won't get ill. Vaccines work in the same way. They contain weakened or dead bacteria or virus, or even just a protein or sugar from the surface. This is enough to convince the immune system that real invader has got in. The same process takes place as when real bacteria or virus invade our body, except in case of vaccines, we do not get ill. Afterwards, if our body ever meets the real thing, our immune system will remember it and get rid of it before we even know it. Vaccine development process is a long, complex and costly process. There are a number of challenges. Here we can see a normal timeline of a vaccine development process, from basic idea to clinical trials to post-marketing surveillance. But we are in an emergency situation. We are hearing a lot about fast-tracking the development process. Here is a graphic representation of fast-track development process. Here, many stages are happening simultaneously. Now let's see each stage in detail. Initial idea. If the initial idea of a particular vaccine seems promising, after elaborate evaluation and research, the idea moves towards product development. The identification of antigens which should induce an immune response is important. Preclinical stage. The aim of this stage is to assess the safety of antigens and select the best vaccine candidate. The vaccine efficacy test is also conducted in this stage. The manufacturing standards are laid down. Clinical stage, phase 1. Phase 1 clinical trials are small-scale trials. Researcher tries to assess two main questions. Does the vaccine generate expected immune response? And is the vaccine safe? The focus is also on to assess what preservative or stabilizer to add so the vaccine does not break down, and whether any adjuvants are necessary to generate a strong immune response. Any preservative, stabilizer, adjuvants that are going to be in the final vaccines must be used in phase 1. The researcher also have to develop tests that consistently show positive results when expected and negative results when expected. Phase 2 trials. Phase 2 trial involves larger trials. Efficacy of the vaccine is assessed. The vaccine safety, side effects, and immune responses are assessed. It involves placebo trial, which means there are two groups. Experimental group, which receive the vaccine, and control group, which do not. The researcher will compare the results of the two groups to have a better understanding of side effects, rate of infection, and more. This phase continues to focus on methods of manufacturing the vaccine, stabilizing the product, determining package vials, etc. Phase 3 trials. It involves thousands of study participants. For example, rotavirus vaccines involve 130,000 participants. It examines how long the vaccines can be used before its expiry, taking into account the transportation and storage conditions. This phase continues to monitor the immune response of the participant and side effect, if any. After phase 3 trials, vaccine manufacturer applies for the license to the regulatory authority, where all the details are included. Phase 4 trials. Vaccine development process continues even after distribution of the vaccine to the masses. Here different groups of study are conducted. One study is conducted in the high vaccine usage area, one involving the whole country, and there are vaccine adverse event monitoring system in place which monitor if a person who received a vaccine had a significant negative side effect. COVID-19 is caused by the novel coronavirus SARS-CoV-2. There are now 7 members of this virus family known to infect humans, and 3 of them have the potential to cause severe respiratory diseases. The two outbreaks preceding SARS-CoV-2 were SARS emerged in the late 2002 and MERS in 2012. Now let's see the vaccine candidates. There are currently more than 200 vaccine candidates under different stages of development. This picture gives an idea about the various types of vaccines and their possible stages. As the processes are fast-tracked, many stages are running simultaneously. The most promising vaccine candidates are mRNA 1273 from Moderna, ADNCOV from CanSino Biological, INO 4800 from Innovo Pharmaceuticals, LVSMENPDC from Shenzhen Genome Immune, Pathogen Specific A APC from Shenzhen Genome Immune, CHADOX1 and COV from Oxford AstraZeneca. From India, Covaxin from Bharat Biotech, Zycovid from Zydus Cadila Healthcare, and Covaxil Serum Institute.